for the opportunity of speaking today. Thank you all for being here. So I'm going to go back in time and focus this time on microbial communities in iron-rich oceans at the onset of the Great Oxidation event. So the Earth has been habitable ever since essentially the beginnings of its origins. This is just a simplified diagram of some periods in time, just to show that even though the Earth has been inhabited ever since we've known of the existence of life 4 billion years ago, different environments existed in which the habitability was probably not the same for different species of microorganisms. For example, at the origins of life, Earth was an anoxic world with an anoxic atmosphere and iron-rich oceans in which the majority of the metabolisms were anaerobic, of course. Around the genomic record and the fossil record tells us that cyanobacteria were able to begin to produce oxygen from water at around 3 billion years ago, or if not earlier. And even though they were able to produce oxygen for a long time, for 500 million years to 1 billion years, oxygen was being produced, but it did not accumulate in the atmosphere. But at some point around 2.4 billion years ago, oxygen production began to ramp up and very rapidly oxidize the globe at perhaps not the modern concentrations of oxygen, but enough to change the metabolic rules of the planet from an aerobic driven to a mostly anaerobic driven to mostly aerobic driven. And now in modern Earth, the majority of the biomass on Earth has an aerobic metabolism. And at the same time, while oxygen was produced, and I'm sure you've recognized this plot from previous talks, iron concentrations decreased dramatically. So what I'm interested in understanding what was the microbial factors, the forces that shaped this period of time of cryptic oxygen cycling, and what made this ramp up so rapidly. So in order to dig a little bit deeper into this, one of the focuses that one of the metabolisms that I'm interested in is the reactivity of oxygen in a ferruginous ocean. So to come up with oxygenic photosynthesis or to produce oxygen in a ferruginous environment is not really a good idea, because oxygen not only will react with cellular components, DNA, proteins, and lipids, and being especially detrimental in an anaerobic world where metabolisms have enzymes that are oxygen sensitive, but under high iron conditions, you have Fenton type chemistry, which exacerbates iron toxicity. On top of that, iron gets oxidized by oxygen, producing solid iron 3, which can also add incrustation and be another reason for toxicity. But there's a problem with this scenario. So in this scenario, it is proposed that cyanobacteria would have had a hard time to overcome iron toxicity, and this could have delayed the oxygenation of Earth. And there's a very important piece missing, though, and which is what I want to focus on, which is other species, other microbes. So around 2.4 billion years ago, which I encircled here in yellow, there were already many ancestral lineages present and diverged. For example, the ancestors of delta proteobacteria and the gammas and betas and pterobacterial ancestors were already likely coexisting at that time. And that means increased number of genes and functionalities and potentially mechanisms to cooperate together. Particularly important could have been proteobacteria. Many of the proteobacteria that we know today are experts in cycling metals, both iron oxidation and iron reduction. So if you include a metal reducer in this scenario, you can have many protective effects by the re-reduction of oxygen or the reduction of iron 3, preventing incrustation. So in order to understand this potential scenario a little bit better, one can study potential early Earth analogs on Earth. The problem with that is that they could be very complex, even though they're more realistic. And also, what I wanted to do is come up with an experimental setting to simplify the conditions and also simplify the number of individuals to control the parameters better and understand the mechanisms that could have been involved in these interactions. Another problem with this setting, the pre-GOE scenario, is that there are no modern analogs. It's really hard to find an illuminated surface on Earth that has an anoxic atmosphere. So what I did is I built a semi-high throughput pre-GOE consortium to explore cooperation on Earth in an anaerobic chamber. So I used 2 model organisms, cyanobacteria, Synechococcus PCC 7002, and strains of Schwannella, of the Schwannella genus, which are metal reducers. Used them either alone or together in pairs in an anoxic atmosphere. And oxygen was allowed to be produced from photosynthesis, but it was allowed to diffuse into the anaerobic chamber. So I conducted these experiments in 10 milliliter volumes inside 20 milliliter wells culture plates. So oxygen would diffuse into a 300 liter chamber, mimicking the anoxic environments. And the treatments were different concentrations of iron 2. So as expected, like previous work had shown, cyanobacteria, as iron concentration increased to 1 millimolar, lost their ability to grow. And it has previously been shown that it's due to iron toxicity. Already at 500, they were showing a lag in their growth. And here I'm showing growth of cyanobacteria in the presence of different strains of Schwannella species. And as you can see, the reverse happens. As iron increases in the presence of Schwannella, most cyanobacterial species, I mean, it's just one species, but in all these different experiments, cyanobacteria is able to grow. And the growth is restored in a way. Schwannella, in the same culture, under higher iron concentrations, the growth is favored too. So there's a relationship between the high amount of iron and the yields and growth rates of Schwannella and cyanobacteria when they're growing together. So when I went and measured iron concentrations, iron 2 concentrations, with cyanobacteria alone in both higher and 500 and 1 millimolar iron 2 concentrations, the iron levels immediately decreased in the first few days, but with Schwannella, the iron levels remained essentially stable, which means that there was not enough oxygen to oxidize iron, probably consumed by Schwannella, or iron could have been re-reduced. But in any case, the mechanisms of protection were related to the availability of oxygen and iron. So among all the different strains tested, Schwannella baltica and Schwannella alga were the 2 that yield similar under high iron or low iron conditions. So to understand the potential reason for these, I incubated each Schwannella independently on its own under different concentrations of hydrogen peroxide, meaning different concentrations of hydroperoxide stress. And so the better helpers, the ones that were able to get Synechococcus to grow almost like in no iron conditions, they were able to resist hydrogen peroxide better. So ROS stress better than the poorer helper. So bringing everything, all these different lines of evidence together, I conclude that iron concentration will impact the interactions between the proteobacteria and cyanobacteria, and this could have driven the community structure in ferruginous and early oceans. Cyanobacteria fitness also increased when there was a metal-reducing partner, so this suggests that the habitability range in a community will change depending on the community composition in a given environment. So this would be important for future habitability tests, whether there's one species or more, will likely change the results. Also, cyanobacteria benefited by co-inhabiting with Schwannella only at higher iron concentrations. So, and again, suggesting that partnerships that may have been important during the Proterozoic or late Archean may be very different than those found in modern environments. So again, analog choice versus experimental setup can yield different results, and important to consider. And also, a cryptic oxygen cycle would have been sustained under high iron oceans by cyanobacteria and proteobacterial partners, which would explain why this would be another biological factor instead of abiotic reasons why oxygen would have persisted at low concentrations or at cryptic levels without accumulating in the atmosphere. So that's the end of the results. And I would like to thank Jen Glass and Chris Reinhardt for their support during these NPP postdoc experience. Thanks to NASA for allowing me to carry out this research, and the amazing people at the Glass Lab and the astrobiology community at Georgia Tech.
Right, we have time for some questions.
Have you looked at the formation of colloids? For example, maybe iron 3 could form a colloid, and maybe other elements, say nitrogen cycling, different oxidation states of another element that might go through iron 2, iron 3 in conjunction with oxygen.
So I put ammonium as a nitrogen source instead of nitrate, which is a typical component of cyanobacteria media. But I didn't look at that, but that may be interesting to see whether reactive species could also be produced by that. Yeah.
Please come to the microphone.
Well, I can do it. Did you see any diel cycling with this at all?
I'm sorry. Diel cycle. So I did not take points. That would be, I was thinking about doing that on a future experiment. I'm going to select one of these Schwannella species and then get more resolution, a more resolved type of growth curve to see if there are changes between the iron and the oxygen levels over day and night cycles. Yeah. This was more like a high throughput to see the range, because even though they're all Schwannella, they're all metal reducers, they do not have the same effect on the cyanobacterial growth, which is also means that it's not a biomass issue. There's other biological components in the explanation of these, other physiological factors.
Okay. Thank you very much.